Okay, in the previous tutorial we talked about inflammation, and in particular we talked about what initiates the inflammation. We talked, here is the 3 stages with the uric acid in mind, but in this one here, the biggest one is this thing here called the inflammasome, and the inflammasome is a very, very complicated molecule. It's made up of many other proteins, and you can see how complicated it is. And what stimulates inflammation, we talked about a range of substances form the inflammasomes, like for example infections, tissue damage, metabolic imbalances like uric acid, etc. So we'll go back here now to the situation where we're going to put it in simple terms of what's happening in the uric acid one. Now the thing is, remember we said we don't want inflammation to continue, because if it continues, it goes from acute to chronic. It can lead to all kinds of other diseases in the body. Here's another lot here which shows you, and I've talked about all those other diseases which are caused by chronic inflammation. We want the inflammation to stop at the acute level, in other words, how long that lasts. So the whole thing here is that we want what we call the resolution of inflammation. So that's based on the formation of these things here called neutrophil extracellular traps, right? Nets resembling tophi, resembling little crystals. So basically the net is this one over here. You can see it. Here it is. It's got a uric acid crystal in it and got these red things, the cytokines, and all those lines there, which is like a net. So it's a neutrophil extracellular trap. Now, and the red ones here, let's start again. What happens? Here shows you a uric acid. I've got it out here. And the red ones are the cytokines, which is interleukin 1 beta, interleukin 1 beta 3, and interleukin 18, and there's a few others. So, the presence of uric acid outside these monocytes or neutrophils stimulates the guys here, the inflammasome, and it starts making cytokines, etc. So here we are, these red ones. Uric acid itself, it actually stimulates the inflammasome, that complicated molecule, but also from inside cytokines are being produced and are released. So neutrophil after the ingestion of monosodium urate. Ingestion, that's what it looks like, and then it forms these things called neutrophil extracellular traps. All right. So if we can stop this happening and we don't get to a chronic phase stage, which is good. So have a look here now into the more detailed mechanisms. Basically, I'm going to be repeating again. The mechanism of chronic tophaceous gout, because it forms tophi, remember? That's what it means, chronic. Now, we don't want it to get to the chronic stage. Neutrophil granulocytes, that's these guys here, neutrophil, ingest needle-shaped MSU, the yellow crystals they go in. And so during low concentrations of neutrophils, the process releases cytokines, the red dots, and it recruits further cells. Recruits more neutrophils, it recruits more other kinds of phagocytes and all other kinds of cells, mast cells that I talked about it before. Now, on the ingestion of these crystals, MSU crystals, neutrophils undergo a specific form of cell death called netosis. Right, there is netosis there forming these nets. Now, when that happens, DNA is excreted during netosis of these densely packed MSU crystals and cytokines. Here they are, and plus those little other fibers in there that call them the nets. But netosis is neutrophil extracellular traps. There's some traps there. Now, the high number of neutrophils deposit allows the formation of aggregated nets, and here we are here, ag nets. which are highly effective in degrading these red dots, which is the cytokines. As it's got here, interleukin-1, it's all those others. It degrades also the MSU crystal and also degrades that net formation of substance in there. So the extruded DNA forms a trap, allowing to neutralize bacteria as well as the danger signals such as MSU crystals. Now here we go, and we look at what's happening is you have these things here called reactive oxygen species. It's not just one substance, it's a whole stack of them, and here they are, negatively charged, and they only last very, very small time, a millionth of a second, but because they are highly reactive, they are very dangerous. They'll break things down. So I've written here, reactive oxygen species, or ROS, with the potential of cellular damage are produced along with the release of energy. And we'll talk about where they are produced. ROS can damage lipids, fats, DNA, RNA, proteins, and which in theory contributes to this physiology of aging. So as people get older, there's a lot of reactive oxygen species, and they start degrading the cells. All right. Now, reactive oxygen species, here. Why is reactive oxygen species produced? Reactive oxygen species are generated during mitochondrial oxidative mechanisms. In other words, the mitochondria produce it. We won't go into that detail. Whole series of chemical reactions, as well as cellular responses to xenobiotics, cytokines, these red ones, and also to the monosodium crystals itself, right? And also these things called xenophobics, this one here. And what is a xenophobic? A lot of other chemicals which are foreign to the body. So, and that's what the word xenophobic, xenobiotic, right? So it is a term used to describe chemical substances that are foreign to animal life. In other words, they're not normally present in the cell. That includes examples such as plant constituents. There's some plant chemicals which are extremely dangerous. Drugs, pesticides, cosmetics, flavorings, fragrances, food additives, chemicals, industrial chemicals, and environmental pollutants. So in other words, these things, these xenobiotic chemicals here, are the ones that can induce the production of reactive oxygen species, which is highly, highly reactive. Now, we'll go back to this diagram here and look at it again, but what happens in terms of what happens to the cell in the gout attack, right? These needle-shaped MSU crystals, yellow, there they are there, are ingested by phagocytes such as monocytes, the neutrophils, etc. The high sodium content of the crystal increases the intracellular sodium concentration. That's the yellow dots there. So, in the cell, before the crystal goes in, the crystal's got the uric acid component, the urate plus the sodium. That's why it's called monosodium urate. And if you look in here, in this diagram here, the concentration of sodium inside is low, okay, and high in concentration of potassium. This is very important to keep the cell balanced to what we call the osmolarity. If you change the concentrations of sodium and potassium, then it affects, water goes in, they swell up. Anyway, they change it. So here we go, MSU crystals uptake. So what happens then is, as you take sodium, monosodium, you've got the sodium component, then you get what we call sodium overload. So you can see here in the yellow dots in here, the sodium level has increased inside, right? And when the sodium levels are increased inside, it causes the influx of water in an attempt to keep it to 300 milliosmoles. I won't go into the details of that. So what happens there is that as the sodium concentration increases, the potassium concentration decreases, and that decrease in potassium concentration, right, is a stimulant also to produce the inflammasomes, all right? And that then the inflammasomes start producing the cytokines in and out. And here is the example of the 3 stages there. These neutrophil extracellular traps are forming there, and then you get this agnets being broken down by reactive oxygen species and other materials here. So, reactive oxygen species, I told you, is very, very dangerous. A type of unstable molecule, unstable means it lasts in millionths of a second, contains oxygen that easily reacts with other molecules in the cell. Here we are, the reactive oxygen species, highly reactive, all right? So what it does, reactive oxygen species, it cracks the DNA, right? No, DNA gets rid of proteins, fat, but it cracks the DNA open and it releases the DNA in the nucleus. Here is the breaking up there, and also the mitochondrial DNA. And so then we have got here an example of the uric acid hyperuricemia, high levels of uric acid, which goes into the cells, releases the cytokines, which I told you before were those the red dots up here. All right, they were the red dots here, and here they are there, releasing a whole lot of them. Pro-ILB1, ILB1, 3, etc., etc. And then that causes the extracellular DNA encompasses that and it forms the net, right? And remember, eventually these are broken down by the oxygen reactive species, this guy here. So the whole idea of the thing is to stop it, to stop the gout process. In other words, to stop the inflammation. Otherwise, that's fructose, but then it can go to other parts of the body. That's the fructose situation. And that's what I'm trying to say here, is that the whole process needs to stop. Otherwise, then we're going to get chronic inflammation occurring everywhere else, and you get organ diseases. We said Alzheimer's. And, we get neurological diseases, cancer. So we want to stop it, and that's what happens through the process that we talked about here, that the ROS eventually causes the agnex, which kills the cytokines, and the cytokines are the ones that can go, and the other cells can go and to other parts of the body and cause chronic inflammation. The resolution of it, that's what we need. We need to stop and not become chronic.